Welcome to The Edge, a program where we spotlight Purdue University faculty, scholars, and innovators, highlighting their careers, scientific achievements, and their vision for their scientific field. My name is Tommy Soares. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology and Infectious Disease. And today I have the pleasure of welcoming Dr. Leopold Green, Assistant Professor of the Weldon School of Biomedical Engineering here at Purdue University. Welcome to The Edge, Leo. Thank you so much for coming here and talking to us today.
Thank you. Thank you so much for having me.
I am so looking forward to this conversation. You have no idea. You have fascinated me. Since I saw you arrive at Purdue, you have fascinated me with your science and your background. I have been a fan and looking forward to being able to speak to an engineer like you about what are you doing in this field of microbiome and how you are really breaking through this field in a way where you're modeling, you're designing different sensors and ways that we can understand what's really happening at the microbe level and microbe host interaction level. This is so fascinating, Leo. Thank you so much for coming. And, I want to hear a little bit about your background, and I'm sure the people that are watching the program would appreciate getting a bit of a knowledge of what is your upbringing per se and experience in the sciences, and maybe we can start there.
Absolutely. Yeah. So I'll start all the way from the beginning. I was born in Las Vegas, Nevada. And yeah, there I was introduced to math and science relatively young and just really enjoyed the concept of both. But I think inside I was really an artist, and I love thinking about design, more structural design and things of that nature. But as I matured through my education, attended Hampton University and actually studied chemistry there. So I stayed more on the science track, but was just always interested in how molecules interacted with each other to form more complex networks. And then that sort of allowed me to expand into my graduate training of molecular programming, which is DNA nanotechnology, and then furthermore into my postdoc with microbiome engineering. So it's been such a fascinating journey watching that that sort of evolve from what I thought I was going to be as a kid, which probably would have been some form of medical doctor or something like that, to now being in the engineering side of biology with medical application. It's been a lot of fun.
And that, what is incredible too is this connection with design and architecture. Tell us a little bit about that. That there must have been inspiration, and I know you're a bit of an architecture buff too. But tell us a little bit about that and how that inspired you to pursue this field in maybe molecular dynamics and scaffolds, DNA scaffolds and structures, and how did that come about?
Well, I think it's just one of those things where I'm interested in commercial architecture, residential architecture as a hobby and personally. But then scientifically, I somehow ended up working with DNA origami, which is one form of architecture, but then also the, I guess, network interaction of host immune response, which in itself is another type of architecture. So it is cool to see them. I don't think I planned it that way, but yeah. So in the world of DNA nanotechnology, we think about what kind of structures we might need to design for a specific application. So we think about, you know, how big does this structure need to be? How might we need to functionalize it? And then we try to test it in a living organism. So there is an element of, you know, classical whiteboard design. You draw it out, sketch it out, think about what it would be, and then you go into a more CAD based design where you actually design put together the sequence of DNA bases to give you your structure, and then finally you experimentally implement it. And it's such a nice process that ties in this art with engineering and biology.
I love that blend. I love that got you going. But I mean, you know, the DNA world must have been like I don't know if it was for you. For me, it was just like, oh my gosh, there's this whole world that we don't even see, but that is so full of these structures and the design of them and their function, right? That blew my mind. It sounds like it had a similar impact on you.
Absolutely. Absolutely.
Um,
Right.
yeah, go ahead. Go ahead. I
Yeah,
don't
yeah.
want
I was going to say it was exactly that. I, joined UC Riverside for my graduate studies in bioengineering. So I made the transition from chemistry into bioengineering. And when I joined the department, I was thinking about tissue engineering. And maybe I mentioned dermatology and medicine, but I wanted to really get into some form of skin, understanding the skin. And but when I joined Riverside, I met my advisor in passing for the first time, I think, earlier in the semester. And she mentioned DNA nanotechnology, and it completely went over my head. And it wasn't until later in the year I did a rotation and I actually got to see what it meant to work with DNA. And I was hooked. It was such a wonderful experience.
and you know what a interesting take on working with DNA and DNA scaffolds or DNA nanotechnology, because most molecular biologists would say, oh, DNA as a way of engineering biology, as a way of engineering, you know, or understanding genetics and just that avenue. But I am so intrigued by your wanting to pursue the structure based and the information that we can get out of that structure. I know you did some work on these cytoskeleton scaffolds and
Sure.
pH dependent sensing. And I mean, can you tell us a little bit about some of that, because that's still pretty breakthrough science,
Sure.
even though I know you published that some time ago.
Absolutely. So in synthetic biology, there is the synthetic cell world where they want to really go from a bottom up approach of engineering a cell that is fully designed by the experimentalist. And one aspect of a cell is the cytoskeleton. And in the early 2000s, Paul Rodeman and a lot of other wonderful DNA nanotech technologists started working with DNA origami. And we decided to use the tile-based nanotube structures because they have the physical shape, they have the cylindrical shape of the cytoskeleton. And it also allowed for us to mimic this dynamic ability of the tubular structures being able to grow and shrink in response to an external stimuli. So we took motivation from the cytoskeleton and we engineered these DNA nanotiles that consisted of 5 short strands of DNA sequences. And these tiles, they interact with each other to form longer tubes in the micron scale. And then we can add DNA or RNA to take advantage of what's known as strand displacement to allow these tiles to break apart, which would shorten our nanotube. And then we can reactivate those those tiles either with enzymatic interactions, and now the tiles can reform to mimic this growth and decay. It was really, really fun work.
I can imagine. I can only imagine. The lab work in doing that must have been just a thrill. I could just see it.
Right.
But the application then is now what? What do we do with these types of scaffolds and the on-off switching that we can
Sure.
detect?
Sure. Yeah, so there similar structures have been used for drug delivery, which is great because now with such dynamical systems, you can envision having these nanostructures as maybe nano cages that can open and close in response to pH or temperature or ligands in the environment. And so I think that is the classic next step for these structures. But I think there's a lot of other cool work that's just fascinating engineering. So using these tubes as walking pathways, and this is also work that's not super new, but still really cool to think about. Having a DNA walker walk along another DNA pathway is really cool, and then having that DNA walker carry and deliver packages to and from different areas is another really cool engineering feat. And to think that this is occurring using DNA, it's yeah, I love it.
It's mind-boggling. Leo, though, could this have an impact on, I don't know, sequencing technologies? Could this have an impact on new types of materials that are smart like this? I know you have a big interest in textiles, but I know your interest in skin is broad. So it's
Right.
I don't want to pigeonhole you in any particular section, but where
Sure,
do we
sure.
go? Like where do we pierce the edge, if you want, of knowledge and technological advance in this particular area?
Right. So I think this is where, there's now this fine line between science fiction and what can really become reality in this space. And so a few areas that I'm looking into, which are sort of long term, is in the wearable space, but somehow functionalizing our textiles with nucleic acids that can sense and respond to um, various chemicals. And because we have, skin and our skin is constantly interacting, we have the possibility, right? So now we're just sort of dreaming, but there's the possibility of being able to monitor what's going on our body with the materials that we are wearing. And I think there are labs that are making strides at building a lot of these components and starting to couple various components. So there's work with DNA, RNA aptamers, which are wonderful sensing uh, um, uh, components that are based on DNA, RNA that can be designed to target any ligand. So you can now envision using this ligand or using these aptamers to detect a specific molecule that gets expressed in the sweat on the body or something like that. I think that's one application. But then the other is just thinking about the microbiome and the human microbiome, but as well as the soil, just being able to understand what's going on in these environments and how these environments are interacting with the host, plant or human.
Yeah, and I just a point of clarification, uh, just give us a brief explanation of this word aptamer that you used, so that
Sure,
people can
sorry.
understand. No. I
Yeah.
love it. I think it's great, but just so that we can, bring in other people that have never maybe heard the term before.
Sure, sure. Yeah. So you can think of an aptamer as a short sequence of DNA or RNA, and they have similar binding properties to ligands as maybe a receptor. So the proteins that sit on the surface of a cell membrane, these receptors they bind with certain molecules. And so the idea is you can have a DNA or RNA strand that can be designed with strong affinity to a specific ligand, whether that's a small chemical, calcium, things of that nature, or it can be a larger protein such as a cytokine.
It's so cool. And that is so awesome. And it's really based on the shape, right? The
Right.
sequence
It's
itself
based on the sequence and
folds
shape.
into a
Correct.
shape that then is able to capture, as you say, the ligand by binding to it, because it has a complementary type shape and is able to bind to it in such a unique way. It's incredible, this world of aptamers and what we're able to start to do with them.
Right.
I think if we could dream of this self-assembly type of world where RNA and DNA are interacting and are assembling into functional units, or allowing us to detect the interactions
That's the goal.
so
Right.
you know,
Exactly.
and
Exactly. That is the benefit of molecular programming with nucleic acids, is that not only can you engineer and build these structures in vitro with a test tube, or inside of a test tube, which is how a lot of the work is initially done, you can then redesign or re-optimize those structures to potentially be engineered to be produced inside of a living organism. And if that's capable, then you can envision that's sort of a synthetic form of like a protein in a way, to where you can begin to functionalize that RNA-based structure to do different things, to encapsulate different molecules. Right? There's so many cool things that can be done with that, and that's where our lab is headed.
that is incredible. And so, do you use computational approaches to design all these molecules and then you test them out in the lab? Is that the approach you guys use?
Correct. So I was trained on a system, or a web-based system called NUPAC, which allows you to input the DNA sequence, and it predicts the binding energy of the bases, whether you're thinking about um, uh, putting two strands together or three strands together. You get to see how they would interact. But since then, there's been so many wonderful platforms. Cat Nano, I think, is one of them that really allows you to get into the details of how these molecules are interacting. And when you're building more complex structures, such as DNA origami, and I hadn't really gotten into the differences between Tilebase and DNA origami, but DNA origami is a much more complex structure. So you really have to understand where all of your staples are binding to give you whatever shape or 3D structure you're hoping to build.
So for people like me that don't really understand those two differences, the origami versus the tile base, can
Sure.
you give us a little bit more? Is tile base mainly two-dimensional, and that's why you need to have a certain set of information, versus the origami is 3D? Is that a big difference, or am I getting this wrong?
Well, so origami is the idea of using um, a scaffold from phage DNA. So this starts with a 3 to 5, 7, as long as 10 kb genome that become that you can then design staples to fold that piece of DNA upon itself to give you, whether it's a two-dimensional A lot of folks Google and they see the smiley face, whether it's a two-dimensional smiley face or um, a sphere. So that is origami, the idea of using the scaffold with staples. Tile-based is usually um, using shorter sequences in maybe 5 or 7 um, oligos. So they might be about 10 to 15 nucleotides long, and you use, or maybe a little bit longer than that, but you use about 5 or 7, and those form a tile, but then those tiles can interact with each other to form more complex, whether those are a 2D sheet or 3D structures.
Gotcha. Gotcha.
Yeah.
Yeah. So cool. And so are we starting to develop nanosensors, biosensors based on these scaffolds, based on these structures, Leo?
Yes, yes. So there's been a lot of biosensors. I think that is probably the first use case of these structures, is to think about using them as potential sensors. Um, and this is really where aptamers have shined, because you can also functionalize the DNA with different fluorescent quencher pairs, which then allows for the ability to have um, to measure interactions and the dynamics of interactions. So yeah,
So neat, so
yeah,
neat. And then you can have interacting DNAs, different scaffolds interacting with different fluorophores, and I mean
right,
you could go and now quantum dots too, and all kinds of other approaches. It's so neat, so neat.
right, yeah,
So what you know, you when you were describing before, you touched on something that I would love to get into because I know you're passionate about, is the information that we can get out of the skin and the skin microbiome. I think most people have heard of the gut microbiome, but I'm not sure that everybody knows that we have microbiome and microbes and all over us.
mhm,
And I think that is something I can you explain a little bit? How is that happening? How is that regulated? What's going on there? What's that
sure.
knowledge?
Sure. So I must admit that I'm not the expert in skin microbiome, but I am so fascinated by what is going on there. So I would say that we are becoming more aware that this interaction between microorganisms and us is quite necessary. And so as we begin to explore the gut and the gut microbiome and the interactions between the gut-brain axes, I think naturally you start to explore what other interactions are there. So of course, the skin is covered with millions, trillions, likely of microorganisms. Um, and surprisingly, these organisms are probably um, most exposed to the environment, right? So we're brushing up against walls, we're touching doors and handles, we're brushing up against people when you're on the train. So there is this constant transfer of the microbiome, but certain parts of the skin are more stable and certain parts aren't. Um, you can also think about just the general makeup of the skin. You have certain regions that are dry, certain regions that are moist. So the diversity of the skin microbiome is quite different, um, which then leads to where I'm most excited about is how does the change in the microbiome lead to chronic skin disease? And then how can we design systems to fix it? And so generally, health and disease is thought to be caused by pathogens or microorganisms or some change in the makeup um, of these microbial communities. Um, but you know, thinking about how we can also make use of the organisms that exist as therapeutics is really the perspective that I'm taking.
Yeah. And
Yeah.
and it's so interesting because you come from this world of structure based, and so I could see that your mind works in a very different way when we talk about these things than a regular biologist would attack the same problem. And it's so fascinating to see you working at the intersection between the microbe and the host and signaling between those. Can you tell us a little bit about some of that work in terms of like the interactions that you're looking for, the dynamic sensing piece,
Sure.
if we could? Yeah.
Sure. Yeah. So I think, there is a lot to be learned about host microbiome interaction. And I think immunologists have been looking at this for quite some time, maybe not directly, but in some form or fashion. And my perspective is thinking about the systems interaction and what kind of network motifs exist within the system, meaning if we think about the immune response and we have an understanding of what a healthy immune cell signaling system looks like, and then we can match that up with what a chronic system looks like in terms of which cytokines are present at different time points of the injury process, which cell types are present, neutrophils, macrophages, things of that nature. And if we have an understanding of what's different between these two, and it maybe even what the failure points are that led the acute to the chronic condition, then we can think about ways of retuning or readjusting the interactions that led to the chronic disease to maybe stop that from occurring.
Yeah,
Yeah.
absolutely. And I think it's your approach that fascinates me is this balance. It's not only what pathogens, but also the dynamic changes in population that you are mapping with
Sure.
with your work. Tell us a little bit about this, because I can imagine that there is a lot of progress in this computational approach for mapping and modeling and understanding the dynamic changes in the population.
Sure, sure. Yeah, so when I started on this idea, I was a postdoc, and I was really just thinking about building a bacterial test bed that mimics a wound injury, which now sounds kind of crazy, but I was just thinking like, what are the dynamics of wound healing? And then how can I engineer a community of E. coli to mimic that? Um, but as I started studying the interactions of what cytokines are being produced, who's producing those cytokines, and the information that I was looking for, at what rate are these cytokines being produced, it was actually quite difficult, at least for me, to find that information. So this is what sort of led me into the systems biology world or the systems medicine, and started realizing that multi-omics data is probably where a lot of this information exists. Still, the question, is there enough of the data? Because I think for the kinds of um, things that I'm working toward is going to require more temporal analysis of omics, right? So not just beginning and endpoint, but every X amount of time over a time course, which I think those studies are starting to be funded now. So I'm excited for that data to come. But once we get that kind of multiomics data, then we get an understanding of more or less the landscape of what's going on in a healthy versus disease state. You get who's present in terms of the bacteria and microorganisms. You get the RNA transcript. You get the metabolites, what communication is sort of occurring as well as the proteins. So once we start to build out a framework for understanding this data, then we can start to use it for how we can build the bacterial systems that might control it.
And so in your lab, do you have computational scientists and molecular biologists as well?
Correct. And actually, so a lot of the students that come probably don't identify with either, but they become both, right? They learn the skills of computational science and data analysis. And I'm actually teaching my students, or I'm encouraging my students to teach me, machine learning and things of that nature, because that's going to be the next iteration of how do we analyze these big data sets. But they get both the machine learning, data science, big data, but then also experimentally the traditional microbiology, cloning. It's yeah, they
That
become pretty well-rounded.
what you're so fortunate to be in a department like biomedical engineering that has such a great and broad a set of resources and people that are similarly minded as you, and have some of that thought as well. And I just love the intersection, because there is so much to be said about the type of trainees that are coming out of your lab with so much computational, but also wet lab techniques and knowledge. It must be phenomenal for them when they come out. Tell us a little bit about, yeah, I know you have some background in working with industry. Have you started to delve into some of that area, or just yet? I mean, I know you haven't been at Purdue for too long, so I might be putting you on the spot. But what are the types of companies that would have you that you've worked with before, and what would be some of the types of companies that you think you would want to start working with in the future?
Absolutely. So I haven't started working with any companies quite yet. I think the the biggest synthetic, or maybe one of the top, would be Ginkgo Bioworks. I think it'll be fascinating to work alongside of them and think about what kind of library of components that we might build, or might be interesting to construct, see if we can do it and optimize it using their high throughput platform. I think things of that nature would be quite exciting. But long term, I mentioned um, the textile route. It would be so much fun to collaborate with any of the manufacturers of silk textiles, or um, soybean based fabrics, things of that nature, because we can begin to take advantage of the biological properties of these textiles, and maybe some of the proteins that might exist in their fiber to activate circuits of the microbiome, maybe the microbiome that's on the textile engineered there, or our native microbiome. So those are the kinds of long-term um, companies that I would love to collaborate with, and start to brainstorm how can we make transformative products.
I love that vision. I think that vision is so cool. And it's part of when I first met you, I think that was what really intrigued me. I was like, oh, I really need to get to know Leo because this is just fascinating. Tell us a little bit about this. I mean, we're talking about everyday textiles, right? The t-shirts and underwear and socks
Exactly.
and whatever it might be, right? So
Correct.
the application can be for the common citizen, but also in the athletics world, right? Sports
Sure,
world. I would
sure,
also think as we're starting to look more and more into space travel with astronauts going up
sure.
there, what type of materials are they wearing? So do you work with materials engineers as well? And have you started to seek out some of those collaborators as well?
Yes, yes. So I'm in the early stages of starting a few collaborations with materials. But I think you're absolutely right. I mean, the opportunity for this is really endless, and naturally we think about athletic wear, but I think there's this big push for understanding health, and with the wearables technology and getting more insight into our daily activities, I think there's an opportunity for us to somehow track what's going on our body in a more holistic manner. But it's going to require a lot of effort.
Yeah.
But I think what's so fascinating about synthetic biology and the community is I'm starting to see that labs are branching out with very unique perspectives. Um, some focusing on uh, interaction between microbes and digital signal transmitters and things of that nature. So it's going to be really really cool to see how all of our expertise and interests start to come together.
Absolutely. I love that. And I think your perspective as a systems biological engineer, for lack of a better term, but I think I see you as that, right? It's this systems approach to the communities, but also systems approach to understand the omics that are of the host and the immune system. That seems to me like a place where really we're starting to make some breakthroughs, or we're going to start making some breakthroughs. You mean to tell us though that there are labs out there looking at how microbes are interacting perhaps with our Apple iWatch type of thing and the microelectronics there?
I am sure. I am sure. I haven't been actively searching this, but I've been brainstorming um, how to improve the detection of what's going on in our body. And I think one of these methods is by quantifying biological markers. So whether these are proteins that are produced in the body or RNA, DNA transcripts, things of that nature. But the question is, how do you access that information? Um, so there has to be a way to transfer what's going on biologically at the molecular scale to the digital signal. And I think there are some pathways there that can be explored, and hopefully we can be involved in that work.
And so are there biological fluids like sweat that are
Correct.
carrying a lot of this information, and that's what you're sampling or starting to get into?
Yes, that is the strategy. I think the question still becomes, is the concentration in sweat high enough or consistent enough for us to trust any measurement? Now, to be frank, I'm not there yet at measuring anything in sweat, but we are building the sensors that should be capable of quantifying this in microorganisms. So we're still a step away from that, but I know labs have started thinking about this. And so, yes, the question becomes, can you just quantify it in sweat? Does it reach the peripheral of the skin and the skin tissue? So can you just measure it at the surface? Or do you need to somehow get access to um, to blood fluids and things of that nature to quantify? And then yeah, from there, I guess you have to make design decisions on which one would be best.
And you know what I love about this line of work is where you're taking it with understanding disease, understanding you touched on some chronic disease. I don't know if you want to touch upon, some of your ectopic dermatitis or that type of, I don't know if through your wound healing work also you're looking at more chronic conditions like diabetes that exacerbate the wound healing process and so on.
Yeah, right. So I think our approach is that we aren't super focused on one particular disease. We're just interested in seeing what information is out there about these various conditions. And so I have a project now where a student is actually looking at two distinct interactions, melanoma, skin cancer, and septic wound healing, which sound like one, two very scary conditions to deal with, but also somehow different, one being a cancer, the other being a chronic non-healing wound. But yet we're identifying that there are some similarities between these conditions within the immune process. So the question becomes, as we look at these differences and look at the similarities, what kind of controller might we need to build that can detect that this that these early signs are actually in fact leading us towards sepsis and not leading us towards melanoma, or vice versa. So those are the kinds of controllers that we're thinking about. And we're no immunologists, so we would love to work alongside experts who really understand the ins and outs of these systems, to help us better design more accurate con controllers that could regulate chronic states.
So neat, so neat. And so, from the wound healing side of things, the the dynamics of the interplay between the immune system showing up at the wound site and then the potential of bacteria that are there and other microbes that are lying around. I mean, that seems to be an interplay that maybe you can expand a little bit on these controllers that you're talking about, and what do you really mean by that?
Absolutely. So, I mean, it's really a battleground, right? I mean, if you think about any of the surface, the mucosal surface that has microorganisms, or the skin, um, there's a lot going on. And I think historically the approach has been regulating population of pathogens. So you have like the antimicrobial peptides, and I think those are fascinating strategies. The one thing that I am always, at least I try to be cognizant of, is when you change the environment or the population by knocking out maybe one of the pathogens, they're likely an opportunistic pathogen, which in my mind means that at some point it became pathogenic, but before that it was probably commensal or useful to the environment. So the design strategy to completely get rid of that organism may also be harmful and could lead to side effects. So with the controller, what we're trying to do is, if the inflammatory level goes beyond a threshold, then we activate a system to bring it back down to healthy levels. And that's very generically speaking, but you can envision maybe you have high pro-inflammatory cytokines, IL-1 beta and TNF alpha, and your organism is able to detect that these concentrations are above that threshold. And when that happens, then it produces a therapeutic that is known to downregulate that signaling pathway. So that's the kind of control that we're trying to implement now. And then beyond that, I think the next step is to really regulate the dynamics of the immune cell response. So who's present, macrophages, neutrophils, for how long, and maybe even their activity. Is it M1 pro-inflammatory or M2 anti-inflammatory macrophage states? And once we can start doing all that, then I think we may have a good chance at trying to improve chronic states.
And Leo, I know from your background though that, and you've been describing, I love the way that you put it. Again, like as a systems approach, you do such a great job of talking about them and talking about the different omics, right? Is there a play for structure-based approaches too, and how we combine those with the other omics to understand? I know from your background, like I could see you being the perfect fit for that type of work.
Yeah, absolutely. I mean, that's exactly where the lab is headed. So now it's the idea of coupling, if I understand correctly, the DNA nanotechnology world with the structural, or with the synthetic biology and the systems biology. And the idea is, if we can take advantage of all of the benefits of molecular programming and being able to engineer these systems to be transcribed in a one-pot reaction, as we would call it, or produced inside of a living organism, then we can potentially functionalize that structure to be a part of the circuit dynamics that we plan to build for the bacterial regulator.
And is it, I mean, with the plethora of bacteria that you would have to kind of sense, is it feasible that we can get there and at least start sensing some of the more dangerous ones that might pounce on us?
Sure. So yeah, I think there are ways to be able to detect pathogens and maybe even distinguish which ones are in the active state of being pathogenic versus maybe when they're in their normal commensal or not harmful state. So there's been a lot of DNA-based neural networks and RNA-based neural networks where you can do this sequence of strand displacement that can sort of decode the upstream information. So that upstream information may be RNA inputs that represent that the pathogen is now becoming pathogenic. And that series of RNA inputs might be complicated, but you can start to build complex networks using RNA systems to be able to break that down. So that's one aspect. The other aspect is, I think, and this is where I'm sort of leaning more toward, is rather than identifying solely that the pathogen is present, maybe using that as one biomarker input. And then a second input might be high pro-inflammatory markers. So now we're detecting that both of these signals are present. And then we might say, okay, there's a critical third signal that if that signal is not present, then we know we need to do something, or some kind of decision matrix like that, right?
Yeah.
And then, yeah, that's when you kick in.
And I, you know, what I find as a big challenge, which I love that you're undertaking, because, you know, why work on something easy, Leo? But your temporal signals that you're chasing. It's easy to go after the spontaneous, 1 second marker. This is what it looked like then. But the dynamically changing environment and set of signals and biomarkers that you are, for lack of a better term, chasing, or that you are starting to look at, that takes a big effort, and I'm wondering, what is the approach there that you're taking so that we can start? Because I'm with you. I've thought about this for so long, how we can make so many inferences and conclusions based out of one single instant that we can see, and how much more we could see if we knew the temporal regulation or whatever it might be.
Absolutely. I think understanding the temporal dynamics is critical to if we want to make a strong effort at remediating this issue of multi-morbidity and chronic disease. But I think this is probably true for a lot of the conditions, or I guess the issues that we're facing in the science community, from uh, I don't know, global warming to soil health and all of this stuff. I think having that temporal data is going to help us understand what's going on, maybe when things are going wrong. So my efforts in this are, we hope to implement, well, we are starting to implement a microfluidic organ on a chip system, which allows us to at least initially be able to tease out how cytokine production changes over a period of time with this unique system when stimulated with pathogen, um, with the pathogen. So then we can measure out how that changes over time and feed that into our model, and as a proof of principle, design the controllers based on that system. And that gives us some confidence, hopefully, that we can be able to detect above or below ranges and things of that nature. So that's my approach. And then I think the long term, what we will need to do is to submit wonderful big team grants where we recruit a bunch of patients of various disease states and healthy states, and let's just analyze them over a period of time. I think that's a difficult effort to put together, but if we can start making strides at collecting this kind of data, it's that alongside of AI and machine learning, I think the opportunities were going to be very beneficial.
Absolutely, absolutely. I think you're right about that. To me, you're painting a picture that's very exciting too, and
I hope
the
so.
combination of these fields and the way that they're coming together is just awesome. And I would ask, what is it about this field that you would want to dispel? Is there a myth? Is there anything that you would want people to know? This is where we're really at. This might be Star Trekky in our
It's
thinking
a
or
good one.
whatever. Is there anything?
That's a good one. I think so. First, I want to start at trying to answer that question, like, where are we really versus where can we be? Like, what's more Star Trekky? And I think that line is so blurry. Synthetic biology is really one of those fields that if there's something you want to try to do, just go find the components and build it and see if it does that, right? So nothing is out of the imagination. Um, I would say with that, but I think to get at the first question, like, what is the myth? And I instantly went to genetically modified organisms and the fear around that. Um, and I think rightfully so, right? I think we as scientists and engineers have to do our due diligence and make sure that whatever we do is responsible. Um, but for the community, I think what's fascinating about the synthetic biology, systems biology, the whole omics, and the combination of all of these fields coming together is that it's been proven with electrical engineering and mechanical engineering that if you have an idea and then you can model it and then you can build it with some um, margin of error built into it, that the prototype aligns with what you model, then you're in the right direction. But that's been at the macro scale. At the micro scale, we're getting really close to being able to do that. So having control, almost complete control over a biological organism that's been designed as a therapeutic or something like that, I think um, will at some point deserve our trust when we demonstrate that the models align with the prototype. So uh, try not to be so fearful of when you hear engineered bacteria as a therapeutic.
Right. I couldn't agree more. I think there's a lot out there that is confusing and scary, but
Mhm.
we are learning a lot also from nature. It was one of my questions that I wanted to ask you is like some of these ideas for the DNA scaffolds and the RNA structures. I mean, there are ribozymes out there. There are all kinds of other structure function relationships between the nucleic acids and the genetic material. I mean, a lot of this stuff we are learning from nature as to how it
Right,
works, right?
right.
Getting some of these ideas and learning how we should be maybe structuring and designing these architectures. But so fascinating. It's so out there for some people that I can understand it's scary. Tell me what types of grad students and postdocs do well in your lab. I know we touched a little bit about the machine learning piece and that they are
Mhm.
challenging you, bringing some of that to the table, which I think it's awesome. I think that the whole group dynamic in your lab must be phenomenal. But tell
Absolutely.
us tell us a little bit about, who thrives in your lab.
Absolutely. Yeah, so my lab is currently composed of classical microbiologists. I think having folks around who understands how bacteria work and what their limitations are, and considering what they want to do versus what we want to do is what makes you a strong experimentalist. Um, so I love microbiologists,
Yes.
computational scientists who think about control theory, um, how to build controllers, how to do sensitivity analysis. Those kinds of students I think are excellent. And anyone who's interested in understanding and exploring the data analysis side to help us infer what might be going on. So this is multiomics. If you're interested in machine learning, I think those three skill sets are fantastic. So having those would be great, but if you're interested in developing them, that is equally wonderful. And yeah, so all students, high school students interested, summer research opportunities. I love hosting and helping out high school students as well. So
Awesome. Awesome. That's so great to hear. And I know that there'll be people listening and they'll be like, I wonder if I should contact Dr. Green and see what I
please,
could do. It's so neat to have you here at Purdue and being able to interact with you and gain some of your expertise too. And I know that your students are probably learning a lot from your wonderful and broad background. Leo, is there anything that we didn't touch on today that you would like to leave us with, or is there a particular topic or question that I didn't ask you that you wish I would have asked you?
oh, no, no, I think this has been fantastic. Thank you so much for inviting me out and allowing me to share my story and my perspective. And if this sparks anyone who's watching to want to have a conversation via Zoom or over tea, I would love to. So please do not hesitate to reach out, email or LinkedIn. I think just a quick Google of my name. It's a pretty rare name, so I think it's easy to find me.
Well, thank you, thank you. I know I will be calling you probably at some other point in time to have a little tea
Please,
and keep this wonderful discussion. I'm
please.
fascinated by the work that you're doing in your world. Thank you so much, Dr. Leopold Green, Assistant
Thank you.
Professor at the Weldon School of Biomedical Engineering here at Purdue University. Thank you for being on the edge, and thank you for telling us about your story of microbiome and the wonderful work that you're doing in your lab.
Thank you. Thank you so much for having me.
Have a great afternoon.
Thanks, you too.
Hey.